It's my pleasure to introduce Billy Brazelton. He comes to us from Blue Marble Institute and the University of Utah. A little history. He got his degree at the University of Washington in astrobiology in 2010. Hoping you correct me if I'm wrong on any of this stuff. So the beautiful part about this is I wasn't astrobiology, but we were both in the University of Washington oceanography department at the time. And he informed me yesterday that my PhD defense was his first PhD defense he got to watch. And that made him continue on to become this amazing scientist. I decided. So Billy and I think we generically knew each other. We're present at the university at the same time. But we both went in various directions. I was not looking at deep sea environments. He continued to write 60 some odd papers on ocean drilling programs, serpentinizing systems, and today he'll be presenting some of that. But then I ended up getting invited as a protein chemist who studied proteins in the open ocean to go to a deep sea seminar, or no, it was a workshop trying to develop new methodology. And I ran across another person we were in graduate school with, Susan Lang. And she and I, shocked to see each other because we also didn't work together. We sat down and had a beer, and I was like, "What are you up to?" And she's like, "I'm going on a cruise with Billy." And from then I was like, "Oh my gosh, I'd love to do proteomics on samples from Lost City hydrothermal vent. Like, that's awesome." So they collect, you hit a horrible hurricane. They collect these amazing samples. And from then, Billy and I started working together, and we just recently got funded. So I requested when Vicky sent out, who should we invite? Billy made the most sense. He's a great example of somebody who's tying geology to chemistry into biology. There's any questions, he will be here afterwards for some time. Very approachable. He's a wonderful man. It's all you.
Thank you, Brooke. That was a very nice introduction. Yeah, as a graduate of oceanography and the astrobiology program, I'm really excited to be here. I was coming back to visit Seattle, see old friends, and also to meet the new generation of astrobiologists. That's been a delight in this trip. So thank you very much for bringing me back. So yes, I'm talking about the Lost City hydrothermal field, which I first started studying when I was a first-year graduate student here a long time ago. And I will, so the site was discovered just before I came here as a graduate student. So I was very lucky to have very fortunate timing to come here and then participate in in the very first expedition dedicated to exploring the Lost City in 2003. The Lost City is located on this giant mountain called the Atlantis Massif. It's the Lost City Atlantis Massif. It's about the size of Mount Rainier, but it's under the ocean. This mountain is on the corner of the Mid-Atlantic Ridge and a fracture zone, which is also called the Atlantis Fracture Zone. Oceanographers name everything Atlantis. These expeditions, when I was a graduate student, were led by Deb Kelly, who is here. Many of you know her. And since then, there have been multiple other expeditions, both with submersibles and with drilling ships. And so that's what I thought I would talk about today, is kind of an update on what has been going on at Lost City and more recently. So in particular, I'm going to talk about two different expeditions that have occurred in the past few years. One in 2018 that I co-led with Susan Lang, which Brooke was just telling a story about. Susan and I were both graduate students here at the same time in oceanography. And then 2 years ago, I was on a drill ship, the Jordy's Resolution, drilling into the Atlantis Massif, the mountain itself. So I'm going to talk about these two projects today. Serpentinization. When I was a student here, everybody was really stumbling over the pronunciation of this word, and we still are. So serpentinization, it's a geochemical reaction where olivine, which is mineral common in rocks from the mantle, is hydrated and oxidized to form this ugly black rock called serpentine. It's called serpentine because it forms that snake skin texture. So I'm a biologist, not a chemist, so I'm gonna go over this in very broad strokes. The reason why we're talking about this in the astrobiology seminar is that this reaction is very common because olivine is very common. It's common in our mantle. It's also common in rocky bodies throughout the solar system. And when it gets wet and it gets oxidized, it releases hydrogen gas. among other things, but for astrobiology, what is really exciting is that it releases hydrogen gas, and hydrogen gas is great food for chemistry in general, and especially biochemistry. So by getting this very common rock wet, we might make food for life. That's the idea. That's what we're trying to test. It's also exothermic, so it releases energy, and it's kind of self-sustaining, and it seems like it might even sustain a hydrothermal system. It releases enough energy. So a place where this might be occurring right now that's not Earth is Enceladus. Here's an artistic rendition of Enceladus, where we have images of plumes being jetted out from the surface of Enceladus. And these plumes might be generated by some kind of hydrothermal system that could have a lot of similarities with the Lost City site and sites like that we can study on Earth. So we think about exploring things like Lost City as practice for sampling things like Enceladus. And what we learned from Lost City could help us interpret results that one day we might get from a place like Enceladus. And so, yeah, just to make sure that point is clear, like the core of Enceladus is probably full of rocks that are olivine rich. And so there's a very good reason to think that serpentinization is happening right now inside Enceladus. You don't know that for sure, but it's a very intriguing possibility. Okay, so that's the astrobiology connection. So now telling you more specifically about Lost City. The Lost City are these chimneys. They form these like skyscraper-like structures. That's why it inspired the name Lost City. But those chimneys are just one expression of a very large environmental system. So at one point I want to get across today is that there are multiple aspects of this system. And over the years, we've been fortunate enough to study multiple aspects of the system. So when I was a student, I was focusing on the microbial communities that live right inside these chimneys, where the hot water comes out, it forms the chimneys, and it mixes with seawater. That's what I spent my PhD working on. There are also the fluids themselves that are coming from below and venting through the chimneys. There's the plume, which is mixing with seawater in the greater ocean. And then there's the bedrock itself. And I'll briefly touch on these things, but I'll be focusing on the fluids and the rocks themselves. And here is showing that there's a gradient of biomass. So these biofilms on the chimneys themselves are very high in biomass. There's lots and lots of microbes living in these chimneys. The fluids, less so. The plume is like seawater, and the rocks are very, very low in biomass. So that has engineering and scientific implications. So here's a Lost City chimney. It's a very unusual among hydrothermal systems in that it's not a metal-rich black smoker, because it is not on above a magmatic system. It's not a volcano. So most black smoker hydrothermal chimneys, they're basically volcanoes on the seafloor. Lost City is not, because it has the serpentinization component to it. So instead of being super hot and acidic, it's only relatively warm. The fluids are usually below 100 degrees Celsius. So it's very comfortable for life. It's very high in pH rather than being acidic. And there's tons and tons of hydrogen and methane because of that serpentinization reaction that I was telling you. And you can see, I don't know if the resolution, you can see here the slime growing on the chimneys. You can actually see the microbes with your naked eye. You don't even need a microscope. And there are animals that live on these chimneys, but they're microscopic. So I always like to point out that you need a microscope to see the animals, but you can see the bacteria with your naked eye. And these are images I produced when I was a grad student showing these are electron micrograph images of the chimneys, and these gray blobs are individual microbial cells encased in a slime attached to these carbonate minerals that comprise the chimneys. So here's the minerals, and directly attached to the minerals are these slimy communities of microbes. That's what I was doing as a grad student here. And we know, well, the other people who have worked on the system, including Matt Shrink, who's also a graduate of astrobiology, that there's interesting biology by comparing these chimneys. Comparing within a chimney, there are young, hot interior zones of the chimneys, older exterior zones of the chimneys. There are also chimney structures themselves that are old versus young, and there are interesting variations in the microbiology and chemistry by comparing these chimney zones to each other and chimneys to each other. And a general takeaway from this is that, not surprisingly, less extreme conditions, more microbial diversity. So on the exteriors that are exposed to seawater, we see a lot more bacteria. And then deep inside the chimney where it's hot and anoxic, we only find a few species of microbes. So not surprising, but that was a lot of work when we were young to figure that out. And so the more recent work that we've done is on, first, so that was the biofilms. We'll come back to this in a second, but now, I think I took out this slide, yeah. So we have done a little bit of work on the plume. That is to say that you can detect the hydrothermal venting in the open in the ocean nearby the chimneys. And this is important for astrobiology, because if you're looking for a hydrothermal system on another planet, you don't know where it is and you have to find it somehow. And that's also how we look for hydrothermal systems on Earth. And so quick point I want to make here is that yes, we can actually detect signs of Lacides venting in the overlying ocean, but it's really, really, really hard. So we have done work on that. You can see that there is an effect on the overlying ocean. You can see hydrogen, for example, and different microbes in the overlying ocean. So velocity is affecting the ocean, but we wouldn't have detected it if we didn't already know it was there. So that's another story for another day. I can talk about that another time. And so I'm going to focus on the fluids themselves. So in 2018, this expedition that Brooke was talking about, we went to sample the hot fluids, warm fluids venting directly from the chimneys. And that's not because we cared so much about the chimneys in this case. We'd already worked on that. As I told you, what we really wanted to do was understand what was going on down here, because this is going to be more astrobiologically relevant. This is going to be more similar to what we'll find in the rocky part of Enceladus, or maybe at the bottom of Europa's ocean, maybe Mars a long time ago, where you've got this big chunk of rock that was recently in the mantle and now been pushed up to form a mountain on the bottom of the ocean. And we want to know what's life down here, because it should be, we're getting a hint of that by looking at the chimneys, but the chimneys are formed because they're mixing with seawater. So we want to know what happens before all that all those serpentinization products are mixing with seawater. So we do that by trying to sample the fluids before they have been greatly mixed with seawater. So in our hypothesis about what we would find if we were able to sample that water deep inside the mountain before it vents, is that there should be a bizarro carbon cycle, I call it. So a weird carbon cycle where organic carbon is abundant because it's so reducing due to all the hydrogen gas. The inorganic carbon is reduced to organic carbon. So you have things like methane, but also formate, and probably other simple organic compounds that are made by the rocks. And because we think that there would be more organic carbon, the species at the base of the ecosystem are actually using organic carbon, metabolizing it, releasing CO2, and then the rest of the community can release the CO2 that was liberated by the biology. But otherwise, they need biology to liberate CO2 because CO2 is basically completely gone in the serpentinization fluids. It's precipitated out as carbonate because of the pH, and it's reduced to organic carbon because of the reducing conditions. So it's a weird, I don't know if there's any other place on Earth where you have no CO2 and you have lots of organic carbon abiotically. That's the environmental starting point. And then biology converts the organic carbon into CO2. So that was our prediction, and that's what we wanted to test by taking these samples. And so Susan and her team designed a fluid sampler specific for this project. You can see here it has a titanium intake nozzle and a temperature sensor in the nozzle so that we can read the temperature in real time as we're collecting the sample from the chimney. And that's important because, as I said, the hydrothermal fluids are mixing with seawater as soon as they come out, and we want to get the purest sample possible that has not been mixing with seawater. And a good way to do that is by looking at the temperature as the nozzle is being manipulated by the robot. So the robot is on the seafloor, we're sitting on the ship, and we're just looking at video monitors and the temperatures reading to see where, if the nozzle is in a good place for taking the sample. So the pilot is moving the nozzle millimeter by millimeter. We're looking at the temperature reading. It'll say a little bit closer, wait, stop, go back. And we do that for hours. And then we finally think it's in a good spot. We take the sample. So that's how we collected the samples to test this hypothesis about the carbon cycle. This might be a good time for me to pause, see any questions so far before I start getting into the weeds. So this is the setup, and how we collected the samples.
Well, how deep do you go?
Hey, Woody.
Yeah.
So the velocity is at the top of the mountain. So that's the water depth is only about 800 meters. But the mountain is about 4 kilometers relief from the trench nearby.
But I was wondering about the sampling drill. How deep is it?
Oh, I see. Yeah, so you can actually see in this video how we've kind of accidentally knocked away parts of the chimney structure by trying to stick the nozzle in. The nozzle is maybe like that big. It's not super long. So this is not a tool good for drilling. We are dependent on finding a good spot. And then we can just put the nozzle in a little place. Yeah, the next project I'll talk about will be drilling. Yeah, this one we're dependent on just finding a good spot.
What was the sampling mechanism here? Does it just kind of shut?
It's a pump. So it pumps in the water, but very, very gently, because if you pump it too much, then you entrain seawater instead of the fluid that you want. Yeah, so you have to do it very slowly and gently.
How old are the vents?
Oh, yeah, good question. There's dating to show that an individual chimney mineral is at least, the oldest minerals they found are 100,000 years old. And the rocks are 1 to 2 million years old. And so the system has probably been there between 100,000 and 1 million years.
I have a kind of general question. So is the serpentine the same serpentine that's found on the surface and affects plant communities?
Yeah. So there are serpentine soils, like especially in California.
Right.
Those are mantle rocks that were serpentinized on the seafloor and then pushed up onto land.
Thank you.
Yep. So it's a very old seafloor. Yep. Exactly. Okay. Great questions. Thanks. So this was the day of this was during our cruise. We're on our way from Woods Hole to Lost City here in the middle of the Ocean. We had a little bit of weather. One of the few. We were lucky to have 4 different hurricanes active at the same time. So we spent most of our time running around the ocean, hiding from hurricanes, and our 12 funded days on site were reduced to 5 days because of the weather, and so we did all of our work in 5 days instead of 12. Which we did by just cracking the whips on the grad students, not letting them sleep. So I have to put that in to make you appreciate that we got this data from 5 days of sampling. Okay, and so we had this very carefully thought out sampling strategy, which is that we would find places and chimneys, places where hydrothermal fluid was coming out that would be high in hydrogen and were directly from the subsurface, and other places where we knew that there were going to be mixing in the shallow subsurface that would have very distinct chemistry. And in the 5 days of sampling, we ended up getting samples that all kind of look the same based on the initial chemistry readings, which were things like temperature, pH, and concentrations of chemicals. And we're like, oh, they're all the same. We had this big idea of getting like hot, high hydrogen, and compare them to cold, low hydrogen samples, and they all kind of look the same. But luckily, a talented graduate student, Carmina Aquino at ETH Zurich, working with Gretchen for Green, and she not only looked at the concentrations, but also at the isotopes of these various chemicals in the hydrothermal fluids that we collected, especially strontium and sulfur isotopes. And to very quickly summarize her results, by using the isotopes, she could infer some flow paths. So this is what I'm actually showing here based on Carmina's work. that she could predict some fluids were had a more direct path through the subsurface and others a more indirect path through the subsurface. A bit based on those measurements, which were not obvious just by looking at the things like temperature and pH. And that was, she was working on that at the same time that I was looking at the results of our microbial community diversity data, where I found that samples from the blue and magenta samples locations were very, very, very different from all the other locations. And just by looking at pH and temperature, I couldn't make any sense of this. The pH and temperature of all the samples were not greatly different from each other, and hydrogen concentrations didn't really quite line up. But this matched Carmina's predictions perfectly. She predicted that these samples had a much more direct flow path from the subsurface than all these other samples, which are more indirect paths, where that hydrothermal fluid was mixing with seawater in the shallow subsurface before it came out and we sampled it. And this was quite a revelation. It really made a lot of sense. Because, or here I'm highlighting that these samples look different from everything else. We're right here. We didn't get good samples of these two sites, I think, because or especially this site right here, because the biomass was too low and we just didn't get enough DNA, perhaps because it was so direct from the subsurface. So not only were those samples very different, when we looked at them, they had species of microbes that made sense for being in the subsurface. So here I'm going to spend just a couple of minutes with some details of microbial diversity data, then I'll come back out, I'll zoom back out again for the non-biologists. So just a few details. So these are those two samples that had the more direct path from the subsurface, blue and magenta. And the species that were most abundant in those two samples are things like methanogens, potential acetogens, sulfate reducers, and fermenters. Metabolisms that would make sense in a hot anoxic subsurface. Whereas species that would require oxygen are not abundant in these samples at all, and are more abundant in those other samples that have more diffuse, more indirect flow paths. So it was actually surprisingly starting to make sense at this point, thanks to these interdisciplinary collaborations. And so my technical expertise is sequencing genomes from organisms without being able to grow them in the lab. So we sequence DNA directly from the water sample, no cultivation required, assemble genomes and try to infer their metabolisms based on the DNA sequences. So that's my technical expertise. I and my students did that, and we searched for evidence in the genomes of how they're living in this environment. Okay, so skipping over how we did that, and I'm just going to give you a couple of results from that work. So these are the individual genomes that we assembled, and here's a little summary of the genes that we found. And first thing I want to say is that this methanogen, which I had first actually studied as a grad student, in the chimneys, we found it in the fluids as well. And it seems like a relatively typical methanogen, actually, as the result of this work, and that it's also capable of nitrogen fixation, which is not totally unusual for methanogen. Second result is that we finally found a sulfate reducing bacteria. When I was a student, we were looking for bacteria that could reduce sulfate with hydrogen serpentinization, and we could never find it. And that's because it was a weird sulfate reducer that hadn't yet been named and annotated as such in the databases. So I just got older and the databases got better, and then we could identify this as a potential sulfate reducing bacteria. So and it's actually the only one of all of the species in all of the samples that we think is capable of using hydrogen and reducing sulfate, which based on Susan's chemistry and other people who looked at the chemistry predicts that's kind of the dominant redox couple on the subsurface is hydrogen from serpentinization and sulfate from seawater. So we found this one species that's very, very abundant in these two particular samples that we think is the key to that part of the biogeochemistry. And the species has been found at other sites of serpentinization as well. So it may even be a general characteristic of serpentinization sites. One more highlight of a microbial species is a few different strains or species of this weird group of organisms that are potential acetogens. So acetogens are like methanogens, but they're bacteria. Instead of making methane, they make acetate. but they have very weird genes. They're not like normal acetogens, and we don't really know what they're doing yet. I'll come back to them in a second though. So those were a few species I wanted to highlight, and now two genes I want to highlight. One is a gene called membrane-bound hydrogenase, a very boring name for a weird gene. And the only point I want to make, here's a phylogenetic tree for those who like to look at trees, but if you don't like to look at trees, only point I want to make here is that we find this gene in not only multiple species at Lost City, but also many species in other sites of serpentinization. So we're starting to find some examples of specific, not only specific species, but also specific genes that are found in multiple sites of serpentinization. So now as a community, we've explored several sites, not just Lost City, but other sites in the ocean and other sites on land. And this is an example of a gene that seems to, that has been found in multiple sites. We have no idea what this gene does. It is called a hydrogenase, but it may not actually be doing anything related to hydrogen. It may be producing hydrogen, it may be consuming hydrogen, but it's found in sites of serpentinization and serpentinization and hydrogen. So that is one potential clue of what life is like in a site of serpentinization. And another clue is related to formate. So I told you that inorganic carbon is reduced to organic carbon, and one of those forms of carbon is formate. And there are lots of genes involved in formate metabolism that we find here, and in particular one that is found in 4 different species at Lost City, and also again, multiple other species and other sites of serpentinization that has this very weird gene that has never been found in anywhere else. So this is a gene its closest relative is a formate dehydrogenase, so something related to formate found in other organisms, like hydrothermal vents in soil. But this particular version of it, these right here, this is actually not, this closest relative is actually quite divergent from all of the examples here, which are all from sites of serpentinization. So it seems like we have found a gene that is specific to serpentinization and is found in many, many different species. So this is bacteria, archaea, So this gene is clearly being transferred horizontally among organisms that live in sites of serpentinization. So again, we don't know what it does, probably something to do with formate, but maybe not. But a clue that we want to follow up on. Okay, so those were a few species and a few genes that we found. This is my summary of the results section of this project. And the message is we found a few species that we think are important for living in this weird environment where you have lots of organic carbon, but not inorganic carbon. We don't know what they're doing, but we have some ideas to do experiments on. Okay, that's that project. Any questions about that? And then I have a second project that I want to talk about. Yeah.
What are your other sites of serpentinization that you're comparing to?
Yep, there's a site in New Caledonia where there are Lost City-like chimneys being formed just off the coast, but the water input there is actually rainwater onto the island. There's a site called Old City. This seems like an old version of Lost City in the Indian Ocean, but actually most of the research is in places like Oman, where there's an ophiolite of serpentinates. There's a lot of good work being done there by Alexis Templeton and her collaborators. And a site in California that I've worked with other people on, where we drilled some wells into the groundwater where there's serpentinization going on. And a lot of these, oh, and there's some weird hot springs in Japan that are serpentinization influenced. And I think, is that all of them? Yeah. Oh, and there's a site in Italy that I've done some work where there are springs, kind of like hot springs, but they're cold and they're also serpentinization influenced. So both seafloor and springs on land. Okay, thanks. All right, so that was our project where we took a robot down and sampled the fluids directly from the chimneys, which is I find aesthetically pleasing because it's mostly non-destructive. We're just trying to find a good spot to sample and we stick a little nozzle in and take the sample. The other project is we took a drill ship, Chinese resolution, a giant a giant drill platform that was, of course, designed for oil drilling. And we drilled into this mountain, the Atlantis Massif that the Lost City sits on. So in our expedition logo, here's Lost City, here's the mountain itself. Here's one well that was drilled many years ago, and then we drilled a new well much closer to Lost City on this expedition 2 years ago. So this is definitely not non-destructive. We're drilling a giant hole into the mountain. About 10 years ago, we drilled some shallow holes into Atlantis Massif, and then 2 years ago, we ran a project where we ended up drilling a well 1.2 kilometers deep into the mountain. So that's what I thought I would spend a little time talking about now. Okay, so this was expedition 399. It was just about 2 years ago we were out there. So previously the only well that had been drilled in a site like this was 1309D, which is kind of on this part of the Atlantis Massif. And there have been some attempts to drill into serpentinites and other places in the ocean, but they were not very successful. So these are other examples of seafloor drilling into rocks. They were only a couple 100 meters deep, and so on this expedition, we were also expecting to only go maybe 100 or 200 meters deep. And we got that deep, and it kept going and 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 going, and it like drilled like sediment. And we never expected to get to 1.26 kilometers deep, but it got that deep in part because of the very good technical skills of the drillers, and also because the rock was so serpentinized. It was so altered that it really was more like sediment than like hard rock. So drilling hard rock is really hard, is really difficult, and that's what all those other expeditions were drilling into and why they didn't get very far. And we were also not expecting to get very far because we also thought we were drilling into hard rock. But at this site, close to Lost City, the rock was actually mushy. And it seems like that's because we were drilling into a hydrothermal reaction zone and not cold hard rock. So the rock was wet and mushy and soft, and it drilled like sediment, and we ended up collecting this 1.26 kilometer core. So the serpentinites look like this. They're full of fractures and cracks like this that are covered in white and mushy stuff, which as microbiologists, we're very excited to see. We don't know that that's biology. It could be just minerals that are hydrated and wet and fresh. But it certainly looks like a nice place to live if I were a microbe. In this diagram, we've highlighted where we took. Hi, Jody.
Hi.
I've color-coded where we took samples for microbiology along the core. So as I told you, we were expecting that we maybe get to 100 or 200 meters. And so we really wanted to make sure we sampled those first few meters well. And so we had a plan for sampling every 5 meters for microbiology. And so we had a plan, take a sample, crush it up in a very, very clean way to limit contamination. And if we had known we were going to get to 1.2 kilometers, we would not have planned to take a sample every 5 meters. We didn't know that. We started, like once we got to here, we were like, okay, yay, we did it. And then we got to here well, maybe we don't need to take a sample every 5 meters anymore. We're like, why stop now? And then we got to here, like, surely it's not going to go any further. Surely we're going to be done soon. And then, yeah. So Jordan is sitting here. She was my partner actually in the rock crushing. And I'll go ahead. These are the tools we used for crushing the rocks. They're these very high-tech things called a chisel and a hammer. It turned out to be the best way to process so many rocks. So we spent 2 months every single day, 12 hours a day, crushing rocks. That's all we did. This is Gordon. He was on the other shift as Jordan and me. Yeah, so that's all we did all day long. But I don't want to skip over these pictures, so these are more pictures of the slimy rocks. Again, I don't know if they're biology, but they sure
So
are
the blue?
lovely slime. Blue?
Go back
In the
one.
back one, yeah, what was that one?
That's just weird lighting.
Okay.
It's very bright white
Oh,
and
okay.
my camera turned into it. Yeah, we called this one the sushi roll. Yeah, okay, so very slimy rocks. Some of the rocks were so slimy that they kept them in the plastic liner and just handed it to me like this, like, please take this away. And then they encased the rest of the core in plastic and hazard tags and took it away, and nobody ever saw it again because they were so scared by how slimy they were. That's a longer story, but I can tell the rest of the story some other time. Okay, I don't have a whole lot of results from this project yet. We're still dealing with the low biomass that I mentioned, but I do want to show off this contamination tracing experiment that we did. So we chiseled the exteriors. Well, actually, Jordan chiseled the exteriors and I did the crushing. And so we sampled the exteriors that we chiseled away for a contamination tracing chemical that we added during drilling. So as we're drilling down, we add a fluorescent chemical. It gets mixed down into the rock as we're coring it. We bring it up and we measure as a positive control that we can detect that tracer in the kind of rubble that just comes up when you bring the core up onto deck. So yes, we can detect this PFT contamination tracer. It's very abundant in our kind of positive control, the rocks, the rubble that comes up when they collect the core and bring it on deck. Okay, so then the exterior that Jordan was chiseling away, we can detect the tracer. Many of the exteriors are actually already very low in the tracer, but many or most of the exteriors have this tracer. And then that interior that is left after we chisel the exterior away, that we're now going to use as our microbiology sample. There are only 2 samples with quantifiable levels of the tracer, and that's what we're using for DNA and proteomics with Brooke and lipids for Jordan and so on. So this was the demonstration that all that work of chiseling and crushing was not for nothing. So our physical labor was successful in removing that surface layer of contamination. That is not all potential sources of contamination, right? There are many other kinds of contamination we still need to test for, but at least that first layer of contamination introduced during the drilling process itself, we can say that we have removed that source of contamination at least. Okay, so this is the work in progress portion of the talk. I am collaborating with the lab in Shanghai, led by Feng Wang, to do the metagenomics. So I visited their lab to help them work out a very low biomass clean room protocol for handling these rocks. Again, there's very, very low biomass, so contamination is a very big concern. Not only is the DNA very low, but these rocks bind DNA very well. So due to the high magnesium content of the rocks, it actually holds on to the DNA. So we need to come up with ways of stripping the DNA away from the magnesium-rich rock. So that's what we're working on now, and they have some preliminary results that I can share with you informally. And then the other component of this is I'm working at the Bigelow lab at the Single Cell Genomics Center, especially Melody Lindsay, Beth Orcutt are my contacts there. And there we're using a technique to identify individual cells that are metabolically active. So the way we did that is that on board, I did an experiment where I took the crushed rock and put it in an experiment with a fluorescent molecule that is redox active. So if there are microbes using any kind of metabolic reaction, they will incorporate this dye into their cells. I freeze them, I send them to Bigelow, and they sort individual cells that have this signal indicating that they were metabolically active when I did the experiment on the ship. So each of these dots is a cell-like particle, and the color-coded ones are those that we were able to sequence DNA directly from that single cell and identify it to, in these cases, the genus level. So this is the one result I have so far, is that this is actually We have data from rocks, but this result I'm showing you here is actually from a fluid sample that we collected from the borehole after drilling. And many of these cells are a genus called Marinothermus, which has been found in other hydrothermal environments. So that is very, very encouraging. But it is aerobic. All known examples, it requires oxygen. And so we need to think about what that means, that in this borehole 1.3 kilometers deep, we have aerobic hydrothermal bacteria. One potential explanation is that the hydrothermal circulation is so fast that even at 1.2 kilometers, you've still got some oxygen left over in the seawater that is coming through that rock, and maybe it's enough to support microbes, or maybe it's a more boring contamination related reason. So we're working through that now. That's the potentially exciting preliminary data we have so far from these samples. Any quick questions about those results? Yeah, Jody.
Yeah, are those strictly aerobic or are they facultative?
They're strictly aerobic.
Strictly, well,
Yeah. Well, I think maybe they can use nitrate or something like that, but
and Polyromonas? Isn't that a curious one to find there?
yeah. Yeah, you would notice the polar monas. Yeah, that one, I don't know. That could be contamination. Yeah. We do not expect zero contamination in these samples. So,
Really,
Yeah.
you've been emphasizing how little you have to work with. Can you give some feeling for how little, this little here?
So we have a lot of rock, right? So we I have a whole freezer full of rock, but then in a chunk of rock, we can get like maybe 1 nanogram of DNA.
Wow.
Which is these days, luckily, you can get data from that. You have to interpret the results carefully. And these results are from an individual cell that came from 10 microliters of the fluid that we collected from the borehole.
Thanks.
Yeah,
How many of the other cell-like particles in this do you think are cells that are dormant, and how many of them do you think are like rock particles that have to be about the right size?
yeah. So yeah, the Single Cell Genomics Center, they're very expert in distinguishing between cells and non-cell particles. And it seems to be a lot of hocus pocus to me, but they're very good at it. So this black area is thousands or millions of individual dots on top of each other, and that's all non-biological particles of some sort. So yes, only a tiny fraction of all of the particles that are detected are active. And actually, I was expecting only a tiny portion of them would be active, and a much larger portion were detected with DNA, but not metabolically active. But actually, I haven't noticed a huge difference in the active versus non-active portion of cells with DNA. But it's still very early. We haven't looked at that many samples yet.
I guess with such low DNA biomass or DNA volume, you're limited to like amplified short read sequencing. Is that correct? Or are you doing other approaches?
We're doing both. Right now, it's mostly short read sequencing, but we're going to do nanopore too.
Cool.
Yeah, it's more difficult. Yeah, you mean with the single cells? Yeah, the single cells.
I
Yeah,
guess for the metagenomes as well.
metagenomes, we're definitely going to try nanopore.
Cool.
Yeah.
And is it still amplified in that case?
We're gonna again try both, yeah, because it's so low DNA, it's possible we can avoid amplification, but yeah. Well, yeah, at the moment, they're trying both. We'll see if we need to use the amplified data. Yeah.
So if there's so few cells, how are you getting this copious amounts of slime?
Yeah, that's a good question. So, two potential answers. One, a lot of the slime is non-biological. A lot of it is very wet asbestos. Asbestos is made by serpentinization. That's where asbestos comes from. Yeah, so there are OSHA stories I can tell about that. afterwards, but sticking to the science. So that's one explanation. The second explanation is that the low biomass comes from averaging all of the cells over a big chunk of rock. And what we really wanted to do, and we already knew that from previous expeditions, and on this expedition, what we really wanted to do is look at those zones that we could tell were slimy. The problem is that we can't use our contamination control procedures on those chunks because there's no exterior to chisel off, right? And so we know they are contaminated, but they're also have the greatest biological potential. So we know there are more cells in those areas, and we're trying to figure out how to sort out which of those cells are contamination.
Do you have cells from the rubble? The part that you said, like, this is the contamination, you can clearly see this is,
yes, we'll have that data.
you don't think you could back out bioinformatically those cells?
Yeah, so we can try to correct for it, but I think that will be unsatisfying, but we'll have DNA sequences from the controls, and that will be, yeah, and we're already doing that. So that'll be a lot of work, but I think that will be the most interesting result, is if we go ahead and sequence the known contaminated samples that are slimy, and then we try to remove the contamination, but that's going to be a very slow project.
Is the majority of the contamination you're talking about, is that like seawater, or is that like once you get on the ship and sneeze on it and stuff like that?
Both, but especially, so the drilling uses the lubricant, which is a very gooey clay material, and it's full of cells.
Yeah.
And that's like, we take a squirt bottle and try to squirt it off, but that's all we can do, right? Yeah, so that's gonna be in there, and we're just gonna try to subtract it out.
Since there's a lot of asbestos, do you think how are the cells protecting themselves from it then?
Well, so asbestos are these really long fibers, and so I see it as a really nice surface area for growing a biofilm. Right, so I think of it as a physical landscape that provides a lot of surface area for growing a biofilm in an area where there must be a lot of fluid flow, because otherwise the asbestos won't be there. So you've got these fractures between rocks, there's fluid flowing through these fractures, you've got minerals depositing that are indicative of warm fluids that are providing surface area. I mean, it seems like a perfect place to grow. I don't know if there are toxic effects of asbestos on the microbes. I think they can just kind of ignore it and treat it as a rock, but I don't know.
Just another silicate mineral.
Yeah. Okay, great. Thanks for all the questions. One last point before I end it, just about so Lost City is famous in its context of the deep sea. It's been recognized by UNESCO as a potential site for recognition in the open ocean. The problem is that there's no legal framework for protecting a spot on the seafloor in international waters. And that really that inability to protect seafloor in international waters became very obvious when the Lost City was included in a tract of seafloor that is awarded to Poland for mining exploration. So this has been in the news a lot recently. There are a few mining companies that are attempting to use the seafloor for mining. It's understandable why that might be something that we want to do, because we are using lots of batteries. Batteries require rare earth metals, and the seafloor does in fact have rare earth metals. I can't imagine how it would be profitable, but that's not my area of expertise. But clearly we should, if we're gonna do this, we should do it in a sensible way. So when Lost City is now somewhat of a poster child for the importance of protecting the ocean from industrial exploitation. And so this is something that I spend just a little bit of my time on now. Yeah, so to be clear, the seafloor mining is not happening yet, but among many of the executive orders that have been released recently, one of them was about announcing the US as being open for business for deep sea mining. And that can happen but outside of even this framework of the UN that was responsible for awarding this, because the US doesn't participate in the UN procedures for ocean conservation. Okay, so on that very happy note, acknowledgements, many students in my lab helped me work on the results that I showed you today, as well as everybody involved in our expeditions to Lost City in 2018 and IODP 399. And because I'm here today, I especially want to thank folks in astrobiology and oceanography here. It was very, very obvious that all of this was made possible by my experiences here. So thanks to all of you. Questions? Oh, hey, Gabrielle.
That was a great talk, Billy.
Yeah,
I'm really
thanks.
intrigued by identifying the serpentization and signature genes, and especially how widespread they are, which is really cool. Is anyone thinking about, are you thinking about overexpressing them, and especially the formate dehydrogenase, and seeing cloning it and seeing like what is it, what kind of enzymatic activity does it really have?
Yeah, that's a really good idea. Yeah, I have a colleague at Utah who does that sort of thing. And so far, his lab was able to express something, a sequence we found in a Lost City metagenome in his lab rat system. And it got to work, and that had to do with DNA repair, which is what he studies. And now, yeah, I'm talking to him about doing things that I care about as well. Yeah, so hopefully in the near future we could do that, but it's not something I'm doing in my lab. So if anybody wants in on that, yeah, let me know. Because yeah, that's what we want to do. We get these sequences, but we want to know what the sequences are doing. So that's why part of what my project with Brooke will hopefully help us out with, and that's another way of doing it. Thanks.
Well, even before you had your last semi-sad thing, I was wondering, is Lost City really unique? No one's ever found another thing anywhere near as good.
Yes, we haven't found anything like Lost City. So in that way, yes, it is unique. But as far as we can understand it, the reason why it is there is this because of this geological process that is definitely not unique and is fundamental to how our planet works. So we would expect to find more things like Lost City. And like I said, there's this thing called the Old City that may have looked like Lost City in the past, and now it's kind of died off. And even if Lost City is unusual in the fact that the water is being funneled in just the right way to make these beautiful chimneys, and it's not common to find these beautiful chimneys, we would still expect that those reactions are happening in the subsurface somewhere that might be difficult for us to notice. But it doesn't mean they're not happening. Right? And that is something we are explicitly starting to study by things like this drilling project that will help us address that question. Like if we find life in these rocks 1.2 kilometers deep, I think that will be an indication that this is a more widespread phenomenon, and that the chimneys were just a lucky indication of what was happening below. Yeah, Jody.
Yeah, so my apologies for missing the beginning. Maybe you already showed the age of the rocks that you sampled. I'm wondering what was the age of the horizon that you sampled where you have identified organisms?
Of the rocks.
Yeah.
Yeah. So it's not like a sedimentary system where it's younger at the top and older at the bottom, right? So these are hard mantle rocks that have been suddenly thrust up from the mantle. And so there will be some variation in age there, but it's a little bit more complicated. But in general, the rocks were uplifted from the mantle 1 to 2 million years ago.
So what was their age? I'm sorry, I didn't.
About 1 million years.
1 million years.
So those rocks have been moving up from the mantle over the past 2 million years. And so we think Lost City as a system has been active for at least 100,000 years and maybe up to close to 1 million in the same location, which is very different from other hydrothermal systems where an individual chimney lives for months or years. but then the plumbing changes and then you find it somewhere else and it's constantly like popcorning around. And as soon as the seafloor spreads, you don't see any activity anymore. Whereas Lost City, this big chunk of rock has been sitting there for about 1 million years, which in geological time is not a huge amount of time, but compared to black smoker systems, for example, it's more.
I was wanting to play a game in my head about scoring that location and the Polarimonus to an ice age.
Aha.
That they were actually deposited at that latitude because we had a snowball Earth or something. But
Right.
I don't see that it matches up that way.
Yeah, I don't know about Snowball Earth, but I think Lost City has been active long enough that the whole ocean, a fairly good fraction of the ocean has now circulated through Lost City.
Yeah, so anything could be there.
Yeah. Some ocean memory there. Any other questions?
Right.
Those are really good questions. Thank you.